And welcome back, everyone. Our next presenter is CytoDyn Inc. It trades on the OTCQB under the symbol CYDY and is a late-stage biotechnology company developing innovative treatments for multiple therapeutic indications using leronlimab, a novel humanized monoclonal antibody targeting the CCR5 receptor. Today, we have a team joining us. We have the president and CEO, Dr. Nader Pourhassan. We have the Chief Medical Officer, Dr. Scott Kelly. We have the COO and CTO, Dr. Nita Ray. And we have the Senior Executive VP of Clinical Operations, Dr. Christopher Reckner. Welcome, gentlemen.
Hey.
Thank you. Thank you for having us.
Thanks for having us.
All right. Well, we are excited for your presentation. I'm sure our audience have lots of questions, so make sure you send those directly to us for CytoDyn. And whenever you're ready, the floor is yours.
Perfect. Yeah, so thank you very much for everyone to be on this call, this emerging growth conference. We are very happy to do our third presentation to shareholders, the old shareholders and possibly new ones. We will be talking about forward-looking statements in all of our presentation today, as always. Please keep in mind that some of the folks on this call don't know CytoDyn very well. So I'm going to take about 1 or 2 minutes and talk about a little bit of history of our company. So bear with me, please. The product that we have, our lead product, is leronlimab, used to be called Pro-140. It had gone through 4 clinical trials at Amarex, the folks who own this product, and it took him about 12 years to do 4 clinical trials, which was 168 patients, and it took the product to 3 weeks of treatment. So the efficacy was shown for 3 weeks. When they asked FDA for phase 3, FDA says you don't have more than 3 weeks of efficacy. So one reason or another, they sold the product, we bought it, and what we have done with the product at that time is we did the following clinical trials, and it took us 7 years to do these trials. And total number of patients that was treated so far with us is 1,445 patients in 7 years. There's 928 more patients pending to be treated in Brazil in 2 different clinical trials, which if we do the number of patients exposed to this product would be over 2,300. So if you just look at the numbers, 168 for 12 years in Progenic, 1,445 at CytoDyn. I'm very proud of our team, and I have asked our key people to be on this call so they can also speak to you of how excited we are with all the activities that we did during these years, with all the challenges. And then one of the main challenges is to raise funds. So you have to have all of these activities happening in a positive way, and it did happen, thank God. And we had $400 million. We raised more than $400 million to do the following trials that you can see. But at the same time, we also have inventory on hand that could support potential sales of $400 million. These are potential values. It could be much higher. This is a conservative number. It could be close to $1 billion if we equate ourselves to the product that's selling for HIV right now in MDR, multi-drug resistance population that we were going for our first approval indication. So the next slide, I would like to talk about the 4 areas that we are very much active. HIV, COVID-19, NASH, and 23 different cancers. One of the cancers is MTNBC, metastasis triple-negative breast cancer. That's the trial by itself. And then 22 other cancers in a basket trial, one of the rare trials that the FDA grants approval for to go forward, and we are one of them. So I want to update everybody on these 4 areas. So what we have is in HIV, everybody's asking, why is this BLA that we have is taking so long? Are we dropping the ball? Do we have the right people? Do we have the right CRO? So I want to get into that. not only mentioning that it's only been 7 years that we've been working on this, and we've been very blessed with very top key opinion leaders believing in this product and helping us. But the BLA delay in combination therapy, we have talked about it before. The dosage difference that we saw that had more efficacy allowed us to expand the use of this product for other indications with a higher dose and be able to be very successful. So that delay was one of the reasons was dosage going to the higher dose to get the optimum dose. But that wasn't the only problem. There was other problems. When you have other clinical trial that you see that has potential value for us, and when you see that, my goodness, you just treated a couple of cancer patients and their physicians are saying things that are very incredible, you have to go forward with those. We can't drop the ball. This is patients' lives. And when COVID-19 happens, we were dragged into this by a doctor we call hero doctor now, Dr. Sidham Raju, Harish Sidham Raju, which we are honored to know that he's nominated to be a board member for our company because he believes in this product. He believes that this is the future of many good things to come for medicine. That's what he believes. That's what the other doctors believe. I'm not trying here to brag about leronlimab. I'm here to tell you the truth and the whole truth. So the BLA got delayed. And before I introduce Dr. Ray and Dr. Reckner and the beautiful combination of the team that we have, I want to show the next 3 slides, and I'm going to come back and talk about Dr. Ray and Dr. Reckner, and they will talk to you about these problems that we have. Before we talk about the problems, keep in mind that the combination therapy in HIV, what's the market value? Is the market value about $1 billion, as we said before in 2019? And this is post-launch, perhaps a couple of years or 2-3 years after that. And what is the market value for HIV in monotherapy? $5 billion? Did we come up with these numbers from the air? No, we did our homework. We asked 2 independent organizations to do their own independent verification. One of them was BioValue. We put their slides in our website. And one of them was Syneos. And I'm going to show you, this is 2019, August. We did this. We evaluated the market size because we want to make a business plan that is sound business plan. And if you see over here, for example, if you look at the middle of the slide, it says MDR, and that's less than 5%. That's what the population we were going to, very small population. And there are big pharmaceutical, Gilead and ViiV being the top 2, that take over the HIV market. But when we talk about all these other cancer and NASH and everything else that we brought, we don't have that problem anymore. We solved that problem. We talked to the big pharmaceutical in HIV with their whole team at that time. One of the things that they said is, your combination therapy is a very, very small market. We're not interested. But monotherapy, you get 90% or more responders. We're interested. We got 90% or more responders, but it's after the first 10 or maybe 20 weeks, which we call induction period, which is another subject, which I don't want to go to it right now, please. Let's keep it just the fact that the big pharmas who took over the HIV, thanks to Gilead and ViiV being 2 of them, done a fantastic job for patients' lives and quality of life. We can do a lot, but we got to go to monotherapy. So why am I saying all this? So let's go back to this slide. Why am I saying this? Because I want to make sure everybody understands that the monotherapy that we started with phase 1 and then got phase 2-3 investigational played a huge role on where we are today. Without monotherapy, patients putting away all their drugs and saying, wow, I have target not detectable. If their viral load was below 1,000 copies per mil, according to published data, they can't transfer HIV. They can't transmit HIV. And if we have a product that gives them a dose a week and they can forget about 4 or 5 pills a day, or some people have 1 pill a day, but still it's annoying to take the pills for some people, that's a fantastic result. We built on that result. But now let's go back to the BLA. Why is it taking so long? Well, the BLA, we have had Dr. Reckner taking control of that, and he found amazing stuff for us about what's going on with our sites, the monitoring, and all the things that he had to do. And then we made the job of BLA to be Dr. Ray, Dr. Nita Ray. And people are saying, why are we changing? We're changing for the better because Dr. Reckner only can work, I think, 23 and a half hours a day. He has to sleep half an hour, I believe. But we'll get to that. So let me read his resume so you know who's Dr. Reckner, just a little bit of his bio. Dr. Reckner joined CytoDyn in August 2020. Prior to joining the company, Dr. Reckner served as a principal investigator in over 100 clinical trials for numerous global pharmaceutical companies, including Amgen, AstraZeneca, Eli Lilly, GSK, Merck, Novartis, and Pfizer. Principal investigator, not just the investigator, principal investigator. He is also the founder of ION Med Systems, a software technology company formed in 2006 with a focus in clinical trial recruitment and management, and a founder of the Center of Advanced Research and Education called CARE, a private research facility formed in 2002. Dr. Reckner was an adjunct assistant professor at Clemson University from 2002 to 2004, a medical director at the United Osteoporosis Centers from 1998 to 2013, and was a private practice from 1998 to 2015, a span of 17 years. Dr. Reckner has a deep background in clinical research. He's also a former diplomat of the American Board of Internal Medicine and is a certified clinical densitometrist. I don't know how to say that. So when we bring him, what did he do? He looked at all the EDC. He's going to talk about what is EDC, electronic data capture. He looked at all the monitoring of the sites. He looked at everything we have, and he recognized that we can do a lot better. But then we give the job of BLA to Dr. Ray for the reason that Dr. Reckner has just enrolled a Nash trial world record, in our opinion, and long hauler that told us we need him to do other trials, and let's see if somebody else can take BLA. Who could take the BLA? This task that has been so difficult. How about a man who built for the first time, he's the one that got involved with this molecule. He knows this molecule better than anybody in the world because he was the one that got involved in Progenic. Let me read a little bit of his bio, and then we'll go to them. In October 2021, Dr. Nita Ray was appointed to additional executive position of Chief Operating Officer. Dr. Ray rejoined CytoDyn in December 2018 as Chief Technology Officer. Prior to joining CytoDyn in 2015, Dr. Ray was Senior Vice President at Progenic Pharmaceutical, Inc. During his 14 years tenure at Progenics, he was responsible for manufacturing, process and analytics sciences, and quality control. He possesses extensive knowledge of leronlimab Pro 140 development. Dr. Ray successfully manufactured the first 10 batches of leronlimab at Progenics under GMP, which was approved by the FDA for use in all clinical trials. Dr. Ray's return to CytoDyn brings 30 years of progressive hands-on experience in strategic planning and execution of process development and manufacturing of biologics, engineered tissue therapeutics, antibody drug conjugates, and small molecule and radiopharmaceutical drugs. He has demonstrated expertise in diverse technology platforms, product development, preclinical, clinical, and commercial manufacturing process and analytical science, quality control, global supply chain, quality systems, and regulatory affairs. Dr. Ray holds a PhD in biochemical engineering from Rutgers University. That's a pretty good resume, I say. That's what I say. And I say that he is capable of doing this job very well. And Dr. Reckner made us to the point where we can hand that to him, and he will take care of these assays. He has done many of these assays in manufacturing, receptor occupancy, anti-drug antibody, and so forth. But with that said, Dr. Ray, let me go to you first. What do you think, where are we with the BLA and the receptor occupancy perhaps? Can you say a few words about where we are with our BLA?
Sure, thank you, Nader. Yes, we are committed to initiate our BLA submission starting next month. So we'll have the preclinical module and CMC module. They will be ready for submission in November this year. We are still working with the clinical module. There are the two key areas that we are still working with and working with a commercial, very reputable commercial bioanalytical service lab, as well as with Dr. Jonah Chacha at Oregon Health and Science University, and to solve the receptor occupancy, that challenge that we had, and FDA made comments, in our original submission. So we're working with very closely with these two organizations, and we believe that, we'll have the BLA will be ready by the first quarter in 2022. That the receptor occupancy will be solved, as well as the anti-drug antibody that FDA has some comments. And we're also working with a commercial bioanalytical lab to resolve that.
Thank you so much, Dr. Ray. Now, in regards to Dr. Reckner, before he joined us, we had people who claim to be very big in this field of regulatory, but they have probably past credibility in publishing papers, or maybe world-renowned cancer people, or whoever they are. And when we had those people who didn't know how to do the regulatory affair with FDA, it took us 1 year to enroll 0 patients in mTNBC, metastasis triple-negative breast cancer. When Dr. Reckner was introduced to me by Dr. Scott Kelly and said, this man is just out of this world in his opinion, and he said he can enroll your CD10, 25 patients that we were needing in less than a few weeks, I almost hung up on him because I didn't believe it, because I never seen it. And he did it. And then I begged him to come and join us, and he accepted. And when he accepted, we saw that he enrolled Nash trial and long haulers, which our shareholders I don't believe they appreciate it as much as what we are seeing. He enrolled 90 patients in Nash trial already. He enrolled all the patients in long hauler 56, whatever, and did the data analysis. And all of these require somebody who knows how to enroll patients, who knows how to go to a hospital, check out what's the bottleneck, fix it, and then get things moving. And if you rely on CROs, and if they don't do their job, then you're stuck. But I want to ask Dr. Reckner to please tell our shareholders, how does the clinical trial gets handled? What is there in a clinical trial that a lot of people come in and say, well, we can get approval and this and that, but then FDA says it takes 12 years, guys. We remember that. And then there is a lot of detail. Tell us a little bit about this process so our shareholders can understand it. Go ahead, please.
Sure, Nader. The process starts with the sponsor working with the CRO to create a protocol. And in the protocol, it's highly strategic. You have criteria that include patients and criteria that exclude patients. The inclusion is basically where you think your medication is going to work. The exclusion is those factors that may confound the protocol. The other part of the protocol that's crucial is being able to look at your primary endpoint. Is that going to work? Are you able to get it at the sites adequately? How do things flow at the sites? And all of that is related to actually not only is this the right testing that you're doing with your protocol, but can the sites handle it, and how does the flow, the operations happen from the site to the CRO to make all this work? Part of that's also incumbent upon the CRO building the EDC system that allows adequate data to be validated when the site enters the data. And there's a two-step process, basically. One is an automated query to any data that may go out of field. And then the other is that the system has to be monitored. With COVID, it's been challenging. We've had off-site monitor visits. But still, you have to oversee that data, and it's an iterative process where the data becomes better and better and better over time. And that's what we strive for with the BLA to get us to where we are first quarter of next year.
So one of the things that you are doing right now, you're transferring all of our extension studies of HIV to a different CRO. Can you talk about that a little bit?
Sure. It's important that we make sure that we follow these extension patients. Some of these patients are close to 7 years, have been on monotherapy, which is incredible. There's 3 trials, CDO1, CDO2, which was drug resistant, and then CDO3. All of these have extended, and we've got a lot of data on these patients that we're looking at to give us clues about how leronlimab is working and how it's beneficial. There is a mechanism of action when we first started with this drug that is simply binding to CCR5 as an umbrella to prevent the virus from entering the cells. But what we've discovered is that binding actually is an immunomodulator. And it's fascinating because CCR5, which is what we bind to, is the actual transmission system of our immune system. And some people's immune systems, for what we're seeing, don't work quite right. I think as a physician clinically treating patients, I wasn't aware of some of these problems that patients were having may actually be due to abnormal immunomodulation. And that's how they were born. Leronlimab binding to this, we're able to see that in some patients that have the transmission stuck in first or second gear can move to fifth or sixth gear. That totally changes what the immune system's able to do. And we see really a lot of that has to do with leakiness of vessels. And I won't get too technical, but it's very exciting what we're seeing with Nash. And we think that that same mechanism of action likely is what's applying to some of the defect in long haulers, but a host of other indications, including stroke, where we're seeing paresthesias and post-stroke fatigue get better, to perhaps implications with this particular immunomodulatory defect with diabetic patients, for example, why some may have worse cases than others. There's a whole lot of research to do.
No, thank you. And you are working with a laboratory yourself directly now that is testing leronlimab for many different biomarkers that I'm very excited for everyone to know about it as soon as you give us a green light to talk about it.
The biomarker was one of the first initiatives. By doing the biomarkers, that helps us be able to select which patients we think would respond. And part of that is also understanding the mechanism of action. And knowing all of that helps us better design the clinical trials. So our upcoming long haulers trial, we've spent a lot of time into that. A lot of the development actually happened through the biomarker lab and what we're seeing actually in Nash as well. And we're just started with the biomarkers. So I look forward to a lot of trials that we can actually accelerate faster by identifying those patients that we can help them better with, that have that predisposition.
Yeah, and Nader, Chris brings up a very good point here, and I think it's something I want everybody to understand. I mean, originally I got involved in the molecule because I wanted to see, it never made sense to me why you would wait for a virus to infect a cell and start making billions of copies rather than block it on the outset, right? Why wouldn't you ever protect healthy cells from viral entry? But Chris brought up something very important, immunomodulation and Nash, and whether you pick your target of PPARP or FXR or GLP or galactin proteins, this is different. This is about CCR5 and immunomodulation. We know that CCR5 is present on hepatocytes. We know that CCR5 is present on stellate cells. Stellate cells are what produce the scar tissue in the liver. So this is really a unique approach in Nash that we think has a high likelihood of success based on immunomodulation. But the same process applies to cancer. The It's about controlling the tumor microenvironment and immunomodulation. That's what's unique about leronlimab. We started with one thing. We just want to protect healthy cells from viral entry. And now we're finding a whole new aspect of different therapeutic indications.
No, that's perfect. Thank you for that. And we want our clinical section of the BLA that had the problem to be submitted by Dr. Nita Ray, the same way he submitted the CMC and had very little comments from FDA. It was all good. So we can't wait for the BLA to be submitted for the combination therapy. But I want everyone to know that we are filing the monotherapy phase 3 protocol, which we filed a long time ago, but we just did not follow through. We are filing not only in US, but we are also thinking about filing that in Brazil. We want to have back-to-back approval when it comes to HIV. We believe our first approval is much sooner than that, hopefully, if we're successful with the enrollment in COVID-19 in Brazil. But we're going to get to that in a minute. But HIV has to have combination and monotherapy. It doesn't make sense if we don't have monotherapy. And that protocol, we're going to get very aggressive. Now that we have over 600 patient data, patients that have gone 7 years and 4 years, we believe that the agency will see that as a positive data. Quite a bit of data was built that would put us in a much better position to go for monotherapy as well as combination therapy. So let's continue with this. So let's talk about now COVID-19. These are the trials that on the left side that we have conducted. The major question right now comes to CD16. Where are we with that with Brazil? And CD17. CD16, we have not injected the first patient yet. Due to whatever that reason, the hospitals have to be comfortable with using the IV. We are using IV for the first time ever since a long time ago when Dr. Nita Ray was involved with Progenic. They done IV use. And Dr. Nita Ray solved all the problems, all the questions that Anvisa had about mixing the product with the IV bag, and he showed that there was no problem. So we should be getting update from Brazil tomorrow. But with regards to CD17, we also have the enrollment going forward. I know everybody wants to know how many do we have? How many do we have? So here's the situation. And by October 18th or so, they have told us, our CRO, the ARO Einstein group, they have said that by October 18, we should have about 20 sites or more on each one of these studies. One is critical, one is severe. Now, if you look at the chart of Brazil, how the curve was going up very high, now it's coming down. They always have problem at some event, perhaps a holiday, where the numbers go really high. If you're set in the hospitals ready to go, we believe we will have days where we will enroll a lot. This happened to us for CD12 in the United States, when all of a sudden we went from 100 patients enrolled to 300 something, and we were able to close the trial. So we will have updates for everybody. Our expectation of timeline is this. We believe that CD16, if we're lucky and are able to get what we are talking about, because this all depends on how many patients will be available, we will be able to get to interim analysis this year. If we are very positive about the results. We talked about that in the last meeting. In regards to Philippines, our orders are getting larger and faster now, and we're very happy with that. But there are work being done with the regulatory department through our folks at the Cairo Pharmaceutical right now that we should have some update within 2 to 3 weeks, is what they told me last night. That could be very, very exciting for us. Now, what does that mean, exciting? We'll find out exactly. I'm hoping that we could have some kind of approval better than just CSP, which is compassionate special permit, perhaps some kind of way that we can sell the product in an easier way. But the orders are going up, and we will keep everybody updated as we go forward. The next thing is NASH. Now, we're very proud of the enrollment, as we said, how fast this got completed. And we want to talk to Dr. Reckner about CD15, which was the exploratory trial. And I'm sorry, this CD15 was our long hauler. Let's talk about the NASH trial. The trial that we just have was 60 patients in 700 milligram and 30 patient open label, 350 milligram. We can close this trial right now. It's completed with the open arm being 350 continue. Am I right, Dr. Reckner?
You are correct.
So if the results are good in a way that we can see substantial improvement in certain population, perhaps we could file for breakthrough designation. However, we don't know the results. It's double-blinded. We are going based upon the animal study results that we had in the past and the work that Dr. Reckner is doing right now in a laboratory and figuring out the mechanism of action. But again, with the Nash trial, we will immediately have a phase 3 protocol, and if we are lucky to have great results, breakthrough designation. This all will happen this year, maybe much sooner. But Dr. Reckner, could you tell us what is your expectation of timelines?
So first, we revamped the Nash protocol, and we were looking at MRI scans. MRI scans are extremely precise, and they're showing the amount of fat, but also the amount of fibrosis. We next, in the protocol, included those patients with a high degree of fibrosis that would have a high likelihood of progressing. So we've isolated the groups to be able to distinguish a better response with medication, a difference between placebo, if you will. We're working with cleaning the data, making sure we're going to the sites, and taking our time to get everything right. We want to make sure all the patients were following the protocol. If they're not, that we record all this, record all the safety information. We expect, to just really do this trial the right way, and hopefully this year, like you said, we'll be able to get really good information.
Perfect. So, and in regards to the next thing which is our 23 different cancer study, this is crucial, and we are seeing results that we believe is very strong, and we are doing breakthrough designation. Dr. Nita Ray has taken over that breakthrough designation application and has done, quite a bit of work getting into the data and making sure that we are able to document all the data. Dr. Ray, you want to talk about the breakthrough designation application that we have?
Yes, we have data on 30 patients on metastatic triple-negative breast cancers, and we are analyzing the data with the Creative Microtech, and we see that there are results that came from in that study, the 30 patients from 3 different studies: the phase 1b/2 trial, TNBC trial. Then 16 patients came from the compassionate use trial, and the 2 patients came from the basket trials, and the 2 patients in emergency IND. So they're all triple-negative breast cancer patients. And we are seeing some results that are very encouraging. And we are comparing that with the standard of care, as well as the sacituzumab govitecan that was originally approved last year, and that was that product is an is an antibody drug conjugate, and that was originally developed by Immunomedics, and they had a fast track designation, and then they had a breakthrough designation, and then during that time they were doing the phase 1 and phase 2, and FDA granted the accelerated approval under accelerated approval provision, accelerated review, and they submitted the BLA while they're still on a phase 1, phase 2 trial that was the single arm trial. And they submitted BLA, and they got a complete response from FDA a year later, primarily because of the CMC-related issues, nothing to do with the clinical trial results, but the CMC-related issues. So they resubmitted the BLA, and they're granted. In last year, they got it was granted, it was approved under accelerated approval provision. And at that time, they were doing the phase 3. It was a randomized trial, and they completed the trial, and the trial was, and FDA granted the full approval this year. So we do see a similar regulatory pathway for leronlimab also. except for the complete response, of course. And we do have the results that we are working on. We think we'll have that, the BTD application will be ready hopefully by next week, but definitely this month. And we are seeing the results are very strong. And in some areas, like for example, like look at the dose response. There are not too many patients, but we have seen the patients at a higher dose, like 525 or 700, is a better response, than at the 350. And also that circulating tumor cell is a good predictor. 73% of the patients who have seen after the first dose, the circulating tumor cells going down, they have a significantly better PFS and OS compared to sacituzumab govitecan drug. That is the currently gold standard for TNBC. So we believe that the results will support a BTD, and we can go from, and we do see that early approval before we even do a randomized trial, that if we follow the same regulatory pathway that Gilead and Immunomedics drug, that would happen.
No, thank you so much, Dr. Ray. And also, I want to make sure everybody understands why we're saying potential 2 breakthrough designation, because amongst our patients, there are 6 patients with brain metastases with mTNBC, and we also have 1 patient who's just breast cancer with met to the brain. So that's 7. We're also going to file, after we file this breakthrough designation, hopefully a week or 2 weeks later, we're going to file for brain metastasis patients, patients that have cancer in breast, but that has metastasizes to the brain. That one, we have 7 patients altogether, and the results are much better than the product that got approved. They had to take the patients in their trial that had brain metastasis out of the data so they could hit the primary endpoint, because that was something they could do per their protocol, rightfully so. But we saw that our brain metastasis patients have done very nicely, and that's why we're going to have the second breakthrough designation. Now, applying for breakthrough designation by the end of this month or next week, the first one, that means 60 days later, around Christmas time, we will be able to get the results, which would be game changer for the company. And the reason Dr. Ray just says the higher dose was what we saw great results. Keep in mind, our higher dose 2-3 years ago, a lot of people say, well, the BLA got delayed, but we opened the door for a variety of all kinds of indications by that discovery. So it paid a lot of dividend for us so far, in our opinion. So the last slide, let's just go over this very quickly. Combination therapy, BLA submission, worst case scenario, end of first quarter of 2022. Best case scenario, hopefully sooner in the first quarter. Monotherapy, we will initiate those trials hopefully soon in Brazil and US. We just don't have any too much. We are meeting with the Brazilian CRO folks, 2 different ones, beginning of next week, not only for that, but also for cancer trials. COVID-19, the long haulers, I want to make sure everyone knows that Dr. Reckner already submitted a new protocol to the FDA, and we have a group of people that are ready to be enrolled. Dr. Reckner, could you talk about that just briefly?
Sure. We're working with the FDA on the trial, Nader. But we have a larger number of patients, a lot more than what we had for the exploratory study. We think we've picked the endpoints very well. And there are so many patients that are wanting to join the trial that I don't think there'll be a problem filling this like we did the last one. So that puts it at results much quicker than what you would see with a normal trial.
Yeah, I can assure you the demand has been absolutely overwhelming for the long haulers trial. So it's very interesting. This morning I was reading, Nader, that there's 240 million cases of COVID. So that means that there's going to be somewhere between 24 to 80 million patients that need help. So obviously we would like to be there to help these patients, but it's a tremendous opportunity for patients and for CytoDyn.
And the way we are progressing forward toward the finish line on these giant indications, I don't know if the shareholders get the magnitude of that. Some shareholders just email me saying that, do we have approval to announce? Do we have approval? Well, we are working on everything, but we believe when this product is able to get the first approval and after that other approvals, there will be tremendous firework for our company, celebration firework. So, okay, now let's go to Nash. Obviously, we just said about Nash. End of November, I'm hoping Dr. Reckner will give us some results. He doesn't like me to say that, but I said it now, so it's too late. And then top-line report, end of December. 23 different cancer, breakthrough designation 1 and 2, both happening this month. Hopefully, that means the results of both will be given to us by the end of the year, hopefully. And then cancer trial in Brazil. Everyone knows that we are in Brazil with COVID-19, and cancer is something we brought up to them. They look at the data, they were very impressed, and they are very much wanting to start very quickly. There's quite a bit of problem with cancer in Brazil, so we can't wait for the beginning of next week to hopefully decide on the CRO, sign an agreement, and go forward. So with that said, can we go to Q&A, please?
All right, great presentation. We do have lots of questions for you all. First one, why is the stock price languishing under $2 per share?
So let me put this in perspective of what the market cap means at $2. $1.60 market cap would be around 1.4 billion. Does that mean great thing for the investors who bought the stock at 30-40 cents? Yes. But there is a lot of investors who bought this stock at $4, $5, $6, $7, $8. So they are panicking about where is the price. The stock will take care of itself as long as the fundamentals are there. Please pay attention what happened when the news came out that Samantha Motit was getting better on COVID-19 in the UCLA hospital with Dr. Otto Yang. When the news started coming out that yes, leronlimab has potential, it did great for our stock. If our activities, as you have seen, the next 6 months, let's just put it 6 months, is going to pan out any one of these things, the stock price, in our opinion, will definitely be taken care of. But we are taking on tremendous responsibility. We didn't drop the ball on cancer, say, let's just wait till we get approval first. We had to add that. COVID-19, we couldn't stay on the sideline because we were brought in. And Nash, Dr. Reckner, single-handedly put us on the map on that one. So let's see what happens with all the news that has to come out. But if you think that we have to get approval next week for the stock to go up, that's not what's going to happen in our opinion.
How is CytoDyn's leronlimab different and better than Regeneron's REGEN-COV and GlaxoSmithKline's sotrovimab and other COVID therapeutics?
Dr. Kelly, you want to?
Yeah, I'll be happy to answer that. So both of those products are really directed toward the spike protein. One of the unique advantages of leronlimab is that it's obviously binding to CCR5 receptor and involved in immunomodulation. So I think those products might have challenges as the variants continue to evolve, and we are variant agnostic. So I think that's a tremendous advantage.
Great, thank you for that. How much revenue will the 1 million vials of leronlimab in your inventory generate, and when?
So when you say how much revenue, first of all, the dollar amount of what you're selling your product is going to dictate that. Ibelazumab is the only antibody which is an IV infusion once every 2 weeks for the patients who have multi-drug resistance. They are selling their product in that field, which is the closest to us, for $117,000 per patient per year. If you just look at $100,000 per patient per year, we got 52 weeks in a year. And if you're giving 2 vials a week, that's 104 vials. If you divide your inventory by that and do the math, it comes out to be a little bit closer to $1 billion worth of product. Can we sell it at that price? No, we might sell it at much cheaper. If we do, then you got $500 million or so. So the estimate that I put very conservatively in the first slide that we presented was about $400 million. Now, keep in mind, we spent 7 years doing all what we have done, spent $400 million of shareholders' money, and we have about $400 million of product probably at the low price even. So that's a pretty good resume, I believe. And then you ask when we're going to get a revenue. As soon as we have emergency use authorization of COVID-19 in Brazil, 700,000 vials will be dedicated to them. And if we are getting cancer, perhaps breakthrough designation, then we have vials. There will be more vials made next year, but we are monitoring everything very carefully. This is a very complex situation and a very beautiful situation, but it has to be monitored very carefully.
And when will you apply for breakthrough therapy designation for NASH?
We don't file for breakthrough designation without seeing the results, obviously. So we have to get the results. We're very hopeful, but we got to be cautious. Look at the data once it's unblinded, and we will let everyone know, like always, what we have.
And can you elaborate on the specific problem you mentioned with the CRO you are separating from on TNBC, and how much of a time delay will result from this?
Yeah, and there is a question that was sent to me that was similar to that, saying that, how come you were so upbeat about the CRO and now all of a sudden this? And why didn't you tell us a little bit at a time what was happening if you were not happy? So when I'm not happy, or when our team is not happy with certain individual in the company or certain vendors that we're working with, we do a lot of analysis. We try to solve the problem. We talk to them. We try to get things moving. To move your company from one CRO who was holding all of our trials and data to another CRO is a very difficult thing to do. We, Dr. Scott Kelly, me, and Dr. Reckner, we went all the way to our CRO's place where they're located, sat there, talked to them, pleaded with them, but there was no indication of we are going to get the data. They, practically we felt like we were being held hostage. And the rate of things getting done in the previous activities was fantastic. It was very beautiful. We were getting a phase 2 monotherapy in 2014, started in February of 2014, went to FDA in December 5th of 2014, and put ourselves in the map to go to phase 3 after Progenics were not able to get to phase 3, and it would have taken 10 more years to get to phase 3, according to their study. So we did fantastic things with this CRO, and we did great things that we're very proud of them. But once we get to the point where the performance goes down, we can't go to the shareholder and say we couldn't solve the problem. We will solve the problem, God willing. But go ahead.
So how do you plan to fulfill any orders if EUA is given? And it seems your inventory is already bookmarked for previous commitments.
So the bookmark is only if we get approval. If we don't get approval in Brazil, and let's just say we get approval for cancer sooner, then we can do whatever we need to do. But to sell product, as soon as you have EUA, there will be multiple choices for us of who do we want to go to distribute. We accepted in the past to go with certain folks, and that would not be a problem. Trust me, anybody in the biotech world will tell you that. If you have an EUA or approval distribution, you will get a lot of opportunities.
So for the NASH trial, you were going to get access to the first 50 or 60 patient data on Monday, September 13th. So do you still plan to update shareholders for their first 50 patients?
No, we're not, because when we stopped working with our past CRO, they locked us out of the data, and Dr. Reckner has done a fantastic job being able to get to the sites and rebuilding those things. So that delayed us a little bit, unfortunately, even though this has to do with patients' lives, we were not getting any breaks from them. So, but the good news is everything's back on where we need to be, thanks to Dr. Reckner, great activities, and we should have that taken care of. Dr. Reckner, you want to say anything about that?
No, all our data was in our possession for the MRIs, which is good, And when we're working with the sites, and they're being fantastic to help us with re-entering the data.
Hopefully results by the end of November. Dr. Reckner, are you okay with that? I'm putting him in a spot. Okay, go ahead.
All right.
Please.
So if MTNBC breakthrough therapy designation is ready to submit to the FDA, why have you not submitted it?
If the MTNBC data, is that what you said?
Yes.
The breakthrough therapy designation, Nader.
Oh, breakthrough. So as we going forward, Dr. Nita Ray has gone to the analysis and have found a lot more positive data out there, especially about the brain metastasis patients. And he's putting it together, and he has top consultants helping him. And I think we don't want to rush something as soon as possible, but before running it by key opinion leaders, perhaps, or other folks, and make sure we have the rock solid data. And that's what we're doing. So hopefully next week, if, as Dr. Ray said, if not this month, for sure, we will have them done.
So you have a phase 3 statistically significant value for HIV. Why are you struggling to get this BLA across the goal line?
So I think we addressed that several times. So perhaps we go to the next question.
All right. Can you discuss the cash position of the company, burn rate, and capital requirements for the next 12 to 24 months?
So in the filing that we just did, we showed, I guess, over $6 million worth of cash. We always said that we will raise money at the time that we think it's the best time for less dilution. And our resume, again, shows by itself what we have done. And I hope shareholders feel comfortable. We have options of doing PIPE, which is private investment, private equity. We have options of doing notes. We have quite a bit of options. We will select the one that dilutes the company the least.
So what is the turn time for new batches of the drug from Samsung?
So the next batch is in the next February. Dr. Ray, is that correct? Next February?
Well, yes, I can address it. That we make Samsung is producing the bulk leronlimab as well as the finished product in vials. So we have made sufficient batches, that will produce about 1.2 million finished products in vials. So we have now today we have more than 700,000 vials in our inventory, and we are making additional batches from the bulk into the finished product in a couple of months. So that will give us about close to 1.1 to 1.2 million vials. Okay. And we have commercial supply agreement with Samsung, and we have the next batches we are producing next year. And that will give us, the bulk product. If we decide to produce the finished product, we can do that. Or if we decide not to finish the finished product, that's to save the stability of the drug. And this bulk product is frozen, so it remains stable for years. So that is up to us, that once we produce next year the bulk product, we are going to make the finished product. And once we make the finished product, we still have stability data go up to 4 years. It remains stable. So we don't have an issue with the stability of the drug. And we had a meeting with FDA this year, actually earlier this year, to discuss our CMC strategy. Because last year's submission, we didn't have Samsung in our CMC package because Samsung was not ready. The process was not validated at Samsung. We had AGC and Ajinomoto in that. So this new submission, we are adding Samsung into it. So that's why it's a much bigger package this year. And FDA had, that we presented what are our strategies, and we had an agreement with FDA, and this is what we are going forward with.
So one quick thing I want to add is when we were first purchased Pro-140 leronlimab, we brought in an ex-Gilead lady who was very knowledgeable about manufacturing. She helped us with getting the product ready for phase 2, and she told me, when you're ready to submit CMC package, you're going to need 100 people to help you. She said, I have done this many times at Gilead, and trust me, you guys are never going to be able to do that. Dr. Ray has done that with his people who he has brought. So I hope people can just Google the CMC packages of, to the FDA for approval of a product and see what kind of workforce you have to bring. And so thank you to Dr. Ray.
I can add one other thing here, Nader, if I could.
Please.
We are submitting breakthrough designation for cancer.
Yes.
And if we have that, and we are going to initiate a phase 2 trial, and that could be the pivotal trial if you get a breakthrough, and if you get the results at what we have seen in the phase 1, phase 2, initial trial. So most of the time that, when a drug is get an accelerated approval, the clinical moves very fast, but the CMC cannot catch up. But we are in a much better situation. This is the same thing happened with the immunomedics. That clinical moved very fast when they got the accelerated approval provision, but the CMC was not ready and they got complete response. We are not in that situation. We have a manufacturing process is fully validated, fully scaled up at Samsung, and we have the drug. And we believe that our approval for cancer, if we get the results in the next trial, they're much faster.
And to add to that, the Humagen, they had a primary endpoint in COVID-19 several months ago, but they couldn't have any product till later on the year, which would be 100,000 vials they were reporting. So to have a manufacturing that we planned 3 years ago, thanks to Dr. Nita Ray and his team, and be able to have that is something I hope people don't take for granted. These are difficult tasks that has been resolved and has put this company in the position that we are, which we believe is the most beautiful position right now. Go ahead, please.
Thank you. So will CytoDyn need a partner to help fund a phase 3 trial for NASH and PASC if trials are approved by the FDA?
No.
Okay. What is needed for uplisting to NASDAQ or the New York Stock Exchange?
My last interaction with them was that you have to have shareholder equity of 5 million or plus 5 million or more. That's no problem for us to have. The second thing is you need to have a stock price of either over 3 or over 4. We like to be over 4. Long time ago, Dr. Kelly remembers this. When we were at 30 cents, people said, how are you going to get the stock over $2 to go to New York Stock Exchange? Are you going to do reverse split? I said, we never will do that. And we got to the point where we are, and we did pass those levels organically. And I am very hopeful that we will pass the $4 ones also to meet that criteria. If we do, when we do, God willing, then we have to do also the situation that we have with shareholder equity value, and then we will have a investor banker in place to raise more funds after uplisting. This is something that they told us they would let us do, NASDAQ. So we are monitoring that, having all of we have already got a lot of documents ready to go. When we get to that point, we will be ready to uplist.
Cool, thank you. So what is the price per vial and how much is each vial?
We don't disclose those things. These are the things that the shareholders should not want me to say it in public. So the team inside it, we can go and negotiate the best deal for us.
And when do you expect interim results for the phase 2 NASH trial?
So as we said, I'm pushing Dr. Reckner to give us some kind of results by the end of November. I don't know if he's going to do that, but he has indicated by the end of the year for sure we'll have the results.
And do you have any updates on the speed at which the current trial in Brazil has been enrolling?
So the enrollment has been very slow, obviously, but the numbers are going down in COVID-19. But again, we have a team of experts that are analyzing, they're meeting with each hospital, and they will be able to solve this, whatever it is, and get us to the finish line. I have total confidence in it.
So you used to mention MS a lot. However, this person hasn't heard it mentioned lately. Any progress on that front? Also, is ALS a possibility?
So the MS is definitely on our map, but Dr. Reckner has indicated dementia, which is related to Alzheimer, and stroke. We had some patients in compassionate or emergency IND for stroke that we saw some great results in 2 patients. But those 2 trials we are working on. But to get MS going also, then again, we need more funding, more people. We're just going to try to focus now getting this to first approval, this leronlimab. Dr. Regner, you want to talk about dementia protocol that you are getting the synopsis ready right now?
Sure, because of what we've seen with stroke patients, having improvements in fatigue, cognitive aspects, and paresthesias, it's a natural fit that multi-infarct dementia, that is thinking or cognitive problems caused by multiple mini strokes, actually would also respond much in the same manner. And since leronlimab crosses the blood-brain barrier and we have receptor occupancy of over 75% in the brain, that fits with this as well too. Plus the CCR5 is upregulated post-stroke. So we look forward to those. We're working actively on those protocols and should have something into early next year.
Yeah, Chris brings up a good point about the neuroinflammatory component. I mean, when you look at everything from stroke to MS to even cancer in the tumor microenvironment in the brain, we think that immunomodulation could help those patients. And the question also asked about ALS, and I've looked at this previously. It's a very difficult motor neuron disease to treat, but the literature is pretty rich in terms of CCR5 antagonism. It's not something we're going to do immediately, but it is on our radar.
Wonderful. Thank you, gentlemen. Do we need another HIV trial for current HIV BLA?
No, not at all.
And what percentage of the HIV population in the trial have become infected with COVID-19?
So in the past, when we asked our folks at the CRO that we had, we were not getting those kind of information. We did hear one time there was nobody at that time. I'm talking about 8-9 months ago, so can't count on that. But Dr. Regner, now that he's going to have these extension studies all under a different CRO, we will be doing a lot of analysis. There's wealth of information here. The patients who generate cancer in the HIV world after 25, 30 years, you get AML and MDS. If nobody developed that's the rate of 25% for that. Dr. Kelly, you want to elaborate on that?
I think it's an interesting point. A lot of the HIV patients have comorbidities, especially with advancing age. And whether you're talking about diabetes or renal failure or liver impairment, if you look at, for example, NAFLD or NASH, it's 30% in the general population, but it can be as high as 65% in the HIV population. So I think that when you talk about COVID and HIV, it's interesting, but we're looking at all the different things, including neurocognitive impairment in the HIV population. And all of these represent a subset of interesting opportunities to help patients within the HIV population. It's not just monotherapy. It's not just combination therapy. It's not just our cure project, but it's all the different comorbidities within HIV that we're going to be exploring. And a lot of that's done through the work of Chris Reckner and the mechanism of action and what we're finding out. And as we link this or put all the pieces of the puzzle together, I think it gets more and more interesting how we can help these patients. And you can make it like a backbone of a regimen to help people with liver impairment, with renal dysfunction, with neurocognitive impairment. So that's something we're going to be looking at as well.
So how has the changes in your company's staff evolved over the past 12 months as multiple new indications are getting pursued?
Well, we have brought tremendous people for us. I mean, we have a laboratory with a very strong people doing work for us. Dr. Regner is working with them. We have an excellent team under Dr. Nita Ray, who he brings people that he needs. Dr. Regner, you want to elaborate anything about the people that you have brought in?
Oh, well, we've got a strong clinical development department. The biomarkers is the key for everything that we're doing, and we're adding people to try to keep up with new trials and new indications that are just exciting to work with this molecule. It's a fun molecule to work with. And
we have a team now that with 4 of us taking care of all the clinical trials, and Antonio Milgrestes, our CFO, doing a great job taking care of all of our other issues, which is quite a bit of lawsuits back and forth and this and that. He's taking care of all of those. I think he does sleep a couple of hours a day, but other than that, he works the rest of it. But the team that he has, I think he has about 10 or 15 people now working for him. And we are just, whatever we need, we will go and get it. For example, right now we need regulatory person for clinical operation. Dr. Ray and Dr. Reckner both are looking into that. And we need the bioanalytic and biostatistician and project manager. We bring all of those. So we will be adding to our team to be able to handle this massive amount of work that we have had positive results with.
So when is the meeting date with the Philippines group going to take place? And did the Philippines request the meeting?
The folks at Cairo Pharmaceutical have indicated that we might be able to have a meeting with them 2 to 3 weeks from today. I talked to them last night, but that's not a sure deal. I mean, we have to just monitor it. I will update everybody as we get information, but please don't come back and say, you said 2 weeks, you said 3 weeks. Let's look at everything that we have generated here, and let's just wait and see what happens. We now are selling product in Philippines under compassionate special permit. That's a big deal for us, and we're going to build on that.
So assuming that you receive one FDA-approved indication, what is the barrier to a second indication or the ability of a doctor to use the drug in other applications?
So they cannot use another application unless we have approval in those applications. That's number one. That's a no-no. Number two is once you get your first approval, you don't need any more manufacturing CMC package to be approved, which is the most difficult part. I think we've got a complete response letter from their last injectable after spending 250 million producing all the data and everything, positive data. The manufacturing failed them and they got complete response letter from FDA. So these are very sensitive issues that we are managing and going forward with it. So, yeah. Next.
Why is CytoDyn no longer supporting right to try? Trials being run in other countries should have no effect on the U.S. patients needing leronlimab.
So when you have a team that as soon as you want to raise a little bit of money, people want to say, why are you raising money? So we're raising money to have more people to do all the tasks. If we want to now do all these emergency INDs that we're getting requested and get the RTT, right to tries, if we do everything for every one of these requests, we will not be able to move our agenda forward in an efficient way. We have to, time ourselves, measure ourselves to be able to get this important product to the people for many indications. So the bigger goal is to do that. So sometimes we just are overwhelmed. We just can't get to those.
Yeah, I mean, I think that the demand has also been very overwhelming with right to try, and there's only so many things we can function on at one time. I can tell you that the demand for the product is certainly there.
Yeah.
So how does CytoDyn think about future fundraising? The FIFE loan appears to be quite toxic. Are there any alternatives?
So whenever the information comes to you from anybody, always I suggest, humble opinion, I suggest that you check it out a little bit, read a little bit. Where is the toxicity of this CVP loan, the John 5, they call it, loan? Mr. John 5 is a fantastic person, which I really like him very much, and he helped us. He is the reason we were able to save some of these patients' lives because we had money, 140 or some $1 million came to us. Who's talking about toxicity? This is a fantastic program he has. You give money to the company has 6 months to pay it back. After that, you can pay back with cash or shares or whatever it is. That way you have access to funds to take the product forward. Products get completely stopped when there is no money. And when you have a fundraising that worked great for us, if he takes $20 million, for example, loan from a company and the company doesn't perform and the stock goes down, yes, this becomes very toxic for them. But we're not that company. We're not. Look at the past of what we did with the loan that we took. We paid back most of it. And we paid it back with a very good terms, and some of it, I believe some cash was paid back. But don't believe on everything that everybody said, especially with this situation that we are in, that people just say whatever they want. It's not true.
So can you explain why some of the tropism tests need to be redone for the HIV BLA? Was there a problem with the assay?
So I think people are, again, spreading rumors that there's no tropism was done with Covance and LabCorp before that. Tropism test is very clearly described what it is to recognize X4 growth in a patient. If it is, then we wouldn't use them. So we have no problem with that. And the ultra tropism test that they have right now, they call it, is 99% accurate for DNA versus RNA. So
explain what assays, in addition to receptor occupancy, are needed before HIV BLA submission, and have RO tests been completed? And if not, when will they be completed?
So Dr. Nita Ray, can you talk about the receptor occupancy aspects of this clinical package?
Right. So we had that, that issue with the receptor occupancy in our original BLA submission. We got comments from FDA, and FDA were very specific what we need to do to get approval. So the method that was submitted, it was not properly validated, not validated at all. And we need to have a validated method to get FDA's approval. So since then, after we got, refusal to file, we have been working with the commercial lab as well as with university in Oregon Health and Science University. And there are some challenges in developing a method, like particularly the CCR5. And we have the best scientific mind, Dr. Jonas Shacha, he's a KOL in receptor occupancy. His lab has done many, many years and has been working on that problem. So he's helping us. And also the commercial, the lab that, they have done many, many receptor occupancies. So it is not only that the method development, that's the issue, that the method needs to be validated. I think that is become challenge if the method is not properly optimized and the method is not robust. So this is one area we are focusing on, and we'll do everything that we can do or can be done. Okay, and we believe that we will resolve that issue. If it is not, we'll go to FDA and say that what are the technical challenges, and we can do certain way that receptor occupancy we have there right now, the method. But if they want to do more on that one, but we are going to address that and how we are going to resolve that. So I think that we are on a right track on receptor occupancy, and we believe that by first quarter next year, we'll have that, the package will be ready with the receptor occupancy that will get, FDA will approve that. Yes.
Yeah, and just one quick thing is about 4 or 5 years ago, the FDA told us that we are not allowed to use this product in phase 3 until we do 9 different assays, I believe. Dr. Nita Ray was in charge, and he had one assay that a lot of people said there's no way to get it done. And his team member, who he has brought a very strong team members, after 6 months, they resolved that and they got that assay done with FDA. So we have a person who deals with FDA and gets it done according to the FDA's guidelines and liking. Maybe other people will talk about, I can do this, I can do that, but you have to see the resume. Have they done something like that and get it done with FDA? Dr. Ray has done that.
Is leronlimab still available through the expanded access program in the US?
No.
So you talked a lot about indications of the goal, and the goal is to get to the large revenue figures as fast as possible. How would you rank the following indications in terms of priorities? Brazil, Philippines, long haulers, HIV, BLA, cancer. So what are the top 3 for you for the next 6 months?
First of all, COVID-19 critically ill population. And I'll also want to add to that we will be filing the same protocol modified. Dr. Reckner is almost done with modifying that protocol to United States FDA very quickly, because there are cases now in America that we could perhaps be able to enroll quickly 200 patients, interim analysis at 100. We will be filing that. At the same time, we might ask the agency for compassionate use once that protocol is in place, then for fee, just something, the same thing we do in Philippines. We're in progress of doing that. So number one, COVID-19 critically ill population. Number two would be cancer. Getting breakthrough designation will put this company in a whole different ballgame. Seeing the results of cancer and talking to the patients, getting the results that we are getting, convinced us that this could be very, very strong in our getting to the approval process. And that's not just one cancer, but 23. So one would be hopefully right away, and then the other 22. The third one would be long haulers. Long haulers, Dr. Chris Reckner is actually working with the FDA to come up with a path that they don't have right now. This is a very new problem for the world. And he's learning a lot from the FDA guidelines and making sure our protocols matches exactly what the FDA says. The protocol has been submitted. So that's number 3. And then 4 and 5 will get to HIV, but NASH would be right where long hauler is. If we have results that are completely fantastic, if it is, then we can have the NASH be also and rank under number 3. But we don't know for sure till we see those data. But the first 3 that I just mentioned is very solid. It's real. Everybody can see the progress we have made. The whole world knows about leronlimab now, practically people in the field. Go ahead.
So for a vial, how many doses does that represent? 350 versus 750?
Each vial is 2.4 milliliter, which is 2 milliliter constitutes of 350 milligram. That's in one vial. Dr. Ray did a fantastic job again to change that from what we had 1.4 milliliters. So we had 0.4 waste. Now it's 2.4 and only 0.4 waste. And two vials will be giving to the patient would be 700 milligram, providing they use only 2 milliliter of each vial.
When will we find out about the 2 new BOD directors once the previous ones leave? Thank you and great work, doctors, from this viewer.
Sure. The two directors that are leaving, I believe you are referring to Dr. Samir Patel and Mr. Alan Timmons. These 2 people have done tremendous work for this company. Just I want to make sure everybody knows that. They came in and we dumped a lot of regulatory stuff. Some of them, that was my fault, perhaps. They said there was some, perhaps, more detailed work we should have done. We didn't, perhaps. I usually like to point finger at the attorney, not at myself. But let's just say whoever fault it was, they had to clean up. They work very, very hard. They work maybe double time to get where we are. And they don't need to do that kind of work. But for them to give us a year of their life like this and helping us, that's tremendous for me. I'm grateful to them. The 2 that are coming in are Dr. Harish Sithamraju and Dr. Lish. What is the last name for Lish?
Yeah, it's difficult to pronounce. Lish Lovu.
So those 2 people, we just had dinner with them, Dr. Scott Kelly and myself in New York a few weeks ago. I cannot wait for everybody to get to know Dr. Harish Sithamraju and Dr. Lish. And Dr. Lish is one of the top key opinion leaders in HIV with a lot of connections to a lot of institutes that can do tremendous research. And Dr. Harish Sithamraju is in Mount Sinai, lung transplant surgeon. And he's the one that told us, you got to be in COVID-19. He had a vision, and his vision came true, and we are delighted to have him. And I can't wait for us to have the shareholders vote for us, hopefully, for these folks and ourselves. And we also have another person, a lady, that we are nominating. And I don't know, I don't recall the last name, but it's Tanya. And she's a attorney, used to be attorney for SEC. So we need to have that kind of talents in our team. And we will bring an audit chairman afterwards, perhaps in December or so.
Yeah, so I mean, I think what's important about this is that it was a very careful and deliberate process about picking the new board members. You know, I've worked with Leash for a long time, and he has just been fantastic. He previously served on our scientific advisory board, and he's one of the top immunologists in the world. But he's also very interested in not only HIV cure, but also HIV and aging, which is an interest of mine, as we talked about before. And Dr. Harissi Demirjiu is not only involved in lung transplant, but advanced lung failure. He's going to help us in multiple different indications. And Tanya Urbach, as Nader said, she's an expert in corporate governance, corporate finance, business growth, investment banking, and an attorney on top of that. So I think it's really going to be a great group of individuals to add to the board of directors.
Thank you. Does CytoDyn have enough funds to make it through the trials in Brazil? And how long does CytoDyn have until they run out of cash?
So this question was asked of me in the last 12 years. Do you have enough money to go 10 more years? We said, no. Do you have enough money to go 5 more years? We don't. Do you have enough money to go 1 more year? We said, no, we don't. What we do is we raise money. And as we need funds, we will raise funds. We have paid the Brazil folks very quickly. We don't wait even a day. We immediately wire the money. We have done that already. And we are very confident we can take care of all of our bills as we have done in the past. Again, $420 or some million dollars should give comfort to shareholders.
How is Dr. Nader's, your mother-in-law doing? How's her cancer progressing? We have a question.
Thank you for asking. My mother-in-law is not doing very well, but she's still alive. She can talk a little bit, very little. She's in a wheelchair. She had 18 lesions metastasized to her brain before she got the leronlimab in November of 2019. She was expected to live 3 months. That's maximum with the brain met of 18 lesions. She's now alive after almost 2 years. So next month it will be her, the birthday of 2 years. So anniversary of 2 years is coming up. We're very excited. I never can wait to go home to greet her. She's the love of our life to me and my wife, and she's living longer than any of the patients. She's a rock star patient for brain metastasis, but everybody keep telling me, don't talk about your own family member. People might think you're just making it up. I'm not. Dr. Scott Kelly has been to my house many times, and he visits with her always.
Yeah, she's a very special woman. Absolutely.
Wow, I'm sorry to hear that, but prayers with her. All right. So how will you finance these trials, and where are we getting the financing from?
So we finance it through the shareholders that we have relationship with in the past. And whenever at this time, people are sometimes reaching out to us, they want to invest. So I don't think fundraising is going to be difficult. It was very difficult when we first started this product and had to change everything that was done with this product in the past and the path to approval, but not at this time. We will raise funds by getting in front of investors and perhaps have PIPE or whatever it is that we decide to do. We let the shareholders know. Our team will make those decisions like we have in the past and perform.
So what is the total projected trial sites for the Biome SA severe and critical COVID-19 trials in Brazil? Where are you currently at? And what is your estimated time frame on full enrollment in the sub-Q severe trial? And what about the IV administered critical trial with the ongoing hold?
So we solved a big problem when we wanted to go to IV, thanks to Dr. Nita Ray and Dr. Chris Reckner. We went back and forth with ANVISA, which is as tough as any regulatory in the world. So having ourselves approved to be there and having maybe 20 to 25 sites by October 18 on each trial ready to go, just the patient comes in and they can get enrolled, is a very great situation. It took us, several months. The number of patients, as I have said, I think CD17, maybe we are close to 20, 25. We'll get the numbers tomorrow. CD16, we haven't enrolled the first patient. As I indicated, this is IV, so the hospitals have to be coordinated for the first patient very carefully. And we will get the results from our CRO, Einstein, and they will let us know. But they have a group of doctors that go to every one of these sites. They're very active. It's not like over here where our CRO was not doing that kind of very important works. And they used to do it at the beginning of our trials 10 years ago, but they were not doing those kind of things. Now we're back to where we believe we can have any problem resolved that comes our way.
Great. So from a viewer, I appreciate that you have enrolled over 560 patients in phase 2b/3 for HIV monotherapy. I wish you would concentrate on scale trials that are the most promising, such as the combo therapy HIV that was completed in February 2018, especially with the lack of size of this company from an employee standpoint. This is frustrating as a shareholder. Please explain why you continue to talk about additional opportunities when we can't seem to get to the finish line with HIV combo therapy.
No, thank you for the question. So I hope everybody knows that 60 or 80 patients that we saved lives of COVID-19 patients and realized that this product has potential in that field was something that we call it, we did not drop the ball. Not that we just expanded. This is something that was called upon us, and because of the results that was seen, papers were published. Dr. Otto Yang's paper was published for 30 of those patients. Dr. Harish Sitamrechu published. Dr. Agresti published. So these are real things, and the fact that we don't drop the ball, we go forward, is the reason that to be happy about and be encouraging us to do so. The monotherapy for HIV has a tremendous market size, 5 billion or so. The other one, the market size was very small. As we try to solve the problem with monotherapy, we realized that the higher dose is the solution. That higher dose is being used right now in cancer, in NASH, in long hauler, in critical ill population, and severe population of COVID-19. So that brought that opportunity for us. Are you telling me that the long hauler, which is post-viral syndromes, probably a bigger indication that could be billions of dollars to this company, was something we should have not done? Or the 23 different cancer trial phase 2 ready to file breakthrough designation we should have not done? And how about the NASH? Maybe we get some very excellent results. Perhaps we should have not done that and just concentrate on what Progenic did. Progenic ran to a big wall when they did that. They went for substance abuse population. They enrolled 2 patients after they sold the product. They enrolled 2 patients in 2 years. And Dr. Jeffrey Jacobson he called me, congratulated me for going in monotherapy path. So we opened the doors, and there's tremendous opportunities. Let's put everything in perspective. How many years did it take us? 7 years. And the number of patients, over 1,400 patients were entered. Did we drop the ball in any of those? And the BLA is being worked on, and all the problems are being solved. And I hope everybody pay attention, because as we get to the approval, then people will be very happy in regards to what we have done in the past. And we will look at all of those at that time again and see what everybody thinks. Go ahead, please.
Thank you for that answer. So how large of a study group will be allowed into the long hauler phase 3 trial? And is it possible to request a breakthrough designation since it would meet the 2 factors required by the FDA?
We cannot ask for breakthrough designation. You have to have a p-value statistically significant. This was an exploratory trial. Our p-value was 0.269, 0.27, which indicated if there is clinical benefit, which it was, the 24 symptoms together dropped 8 points in placebo, and it dropped 16 points in the leronlimab arm, 100% better. Clinical outcome was there. We believe that's a very beautiful thing for us. But now we have to do a trial. What trial? Dr. Regner has worked with the FDA on that and has submitted a protocol. Dr. Regner, you want to say anything about the protocol that you submitted?
Well, with FDA guidance, I think we've got a very good package that we submitted to them. We learned a lot from the CD15 trial. We learned a lot based on the biomarkers and what patients would respond and what co-founders or confounders would potentially interfere with the study to pull those out. We've also got a 350 versus 700 dose. So we feel very strong about it with an interim analysis that we think that we can get in.
So how many patients were in that protocol that you submitted?
225.
So 225, can you enroll those very quickly?
Absolutely.
I trust
Absolutely.
you because you've done that before. So, but please.
We've got, there are about 500 patients on lists at different sites, different states. So we just want to try to help them, Nader. You know, this has been, I get the frustration of the shareholder with the BLA. We're not losing focus with that. We had some things to get straightened up, and we are now on it. You know, Nita Ray, taking care of the assay development, has helped me focus on working with the clinical and non-clinical. And so we're tag teaming it. I feel confident he's got our back for the assay development with his team. And he tells us when he needs the labs, and we get them for him.
No, absolutely. And we feel very strong. If we have a next trial green light and enroll this year, if we're able to have the CRO that Dr. Reckner would select, which is in process of doing, then if we had primary endpoint with that one, then I'll be very hopeful for a breakthrough designation. Yes, but other than that, at this time, no. Go ahead, please.
So how do you justify HIV combo dosage using HIV monotherapy when combo therapy uses an antiviral that suppresses CCR5 expression on T cells?
Well, if you look at 1 cubic centimeter, it's just this much, of leronlimab, there's a so-called Avogadro number. It's 6.23 times 10 to the 23. That's how many Pro-140 you have in there. And if you look at the T cells, 10 to the 10, according to the literature out there, 10 to the 10 numbers. Even if there is 1,000 CCR5 on each one of them, then there's 10 to the 13. But you got that 1 milliliter has that many leronlimab. So when you do the numbers, you got about 1 million, Pro-140 leronlimab for one cell. So I don't think there's anything wrong to give that in combination to the drugs that, yes, they do help the CCR5 to be augmented, perhaps.
So just to clarify the particulate issue in Brazil, was Anvisa just verifying there would not be an issue with particulates, or was there an actual particulate problem mixing leronlimab with the IV solution?
No, there was no problem. Dr. Nita Ray was in charge. Dr. Ray, would you like to talk about the assay that you did?
Sure, yes. So we did, add leronlimab, this is the subcutaneous formulation, into IV solution that was in an IV bag. So when we did that, so we wrote a protocol first, how we are going to do the test, and submitted to Anvisa, and Anvisa approved that protocol. So then we contract out to a CRO to do this testing. So one of the tests that is called particulate tests, and there are several other tests, and all other tests, everything in terms of potency, that is pretty much as very close to control. So there's no issues. On the particulate test, there is there are two different tests. One is called it's a light imaging test, and sometimes the test cannot distinguish from the particulate to the bubbles. So when you mix that, some bubbles will form, so invisible bubbles. So that lab was not able to do that, in a second test. That's a microscopic test. So we went to another lab, and they can do both the imaging test as well as the microscopic test. And we found that the microscopic test verified that there is no particulate issue. So it is as good as the control. So we submitted that to Anvisa, and Anvisa approved it. So that's what we can go forward with the IV formulation, and we don't have any issue with that. Right.
Thank you.
So why has this SEC not yet authorized your last two S3 filings?
So according to our CFO, they have. We are giving those kind of S3 shares to our vendors, the people that we got notes from. But if it hasn't, if there is some S3 that we didn't get yet, we're not trying to get S3. S3 is registered shares. We don't want to raise money with registered shares that people can sell right away. If we raise funds, we want to be locking the shares for at least 6 months, which is the normal course of shares not being available. We believe our company in the next 6 months can do things that we've been working very hard in the last 7-8 years to get it to this point. And the next 6 months, whoever invests in our company, I believe it's a great investment. So we're going to lock the shares for 6 months. So we don't need this.
What is the effect of chemo on leronlimab? Has leronlimab been studied with chemo plus radiation and surgery, as well as leronlimab with radiation plus surgery?
Dr. Kelly?
Yeah, no, I'd be happy to comment on that. So we don't see that there's an effect of chemo on leronlimab. What we believe is that there'll be a synergistic effect with leronlimab and chemotherapy. Chemotherapy can cause increased CCR5 expression. What we hope to eventually get to is the point where, as people know, chemotherapy the side effects can be almost unbearable. And what we'd like to do is add on leronlimab to chemotherapy, be able to reduce the dose and have a synergistic effect. And that would be the ultimate goal with chemotherapy. I think it also can be combined with antibody drug conjugates, PARP inhibitors, checkpoint inhibitors, et cetera. So I think there'll be a synergistic effect. And there's also the possibility of monotherapy. We have, in certain trials, been able to combine it with chemotherapy, and we have not seen a drug-drug interaction, which is very encouraging. It can also be combined with radiation.
And one thing I should add is that somebody asked me in the email, I believe it might be one of the questions, that when you're using leronlimab with chemotherapy, how do you know it's chemotherapy working, not your product? So there's something called historical data. When we did our monotherapy in HIV, we couldn't have a placebo arm where the patient's viral load goes through the roof and they get sick and God forbid they die. We couldn't have that. So we go with the historical data. Historical data is well-documented, 1,000, 10,000 patients who, when they don't take their medication, is proven that it will take 2 to 3 weeks for their viral load to break through and get to a dangerous position. That's called historical data. With the chemotherapy, for example, with the MTNBC, carboplatin was not being able to be used consistently for 6 months in patients. That's why top key opinion leaders told us, if you use carboplatin in your MTNBC with leronlimab, and if you only have 5 patients that can go for 6 months without changing their chemo regimen, you got a breakthrough designation. And that's how we leave this MTNBC that we have right now. It's very strong because we're looking at historical data of, I believe, 12,000 patients data.
So how much efficacy data is needed for registration of long haulers?
Well, that's something the FDA has to comment on. Well, obviously, you had a primary endpoint in a phase 2 of 200 some patients that Dr. Reckner just mentioned. Then we feel great to go to the FDA. Dr. Reckner has done a fantastic job with getting the right information extracted from this small trial that he conducted so quickly. Now that data is in work to be able to make the next trial. Same thing with Brazil trial. We learned so much from our CD12. And if we had the right folks at that time to help us and give us the data that some people held data from us, wouldn't give us the receptor occupancy. I don't want to talk about those people, but when they didn't, they hurt us. They hurt our chances of hitting our primary endpoint because they took our data to the laboratory and would not give us the result. Those things are not going to happen now. We are in good hands, and we believe we're going to have quite a bit of beautiful future ahead of us.
Good. Are there any further trial inquiries coming in from India or the Philippines?
Nothing for now.
And what is the delay for the first patient IV for CD16?
I will know that tomorrow after meeting with the Brazilian folks, and I will be very happy for our team to report to the shareholders immediately.
And can you give an update on the number of patients who are currently enrolled in CD16 and CD17?
CD16, 0, because we're trying to get the first patient. And CD17, I believe we might be at 20, 25 right now. Again, they're enrolling hospitals. They had like 3, 4 at the beginning. Now they should be at 20 by October 18th. That date that I'm telling everyone is where we feel very comfortable having quite a bit of hospitals, and we will be adding to those. But then hopefully the enrollment should pick up.
Is the age issue that made CD12 fail be addressed in the Brazil trial?
Absolutely. That's one of the things that we learned. And again, it was something that you cannot tell when at the beginning of the pandemic, you had no data about these things. So yes, absolutely.
Any plans on a trial on post-stroke depression? Pfizer's testing this with their CCR5 antigenist Maraviroc based on recent study results. Tabasco, Tel Aviv, Swarovski Medical Center show outstanding potential results. Sorry, I botched that question.
Yeah, we love it when Maraviroc does beautiful. God bless Pfizer and Viv for that. When they do, that gives us a chance to immediately go to FDA and ask for phase 2 because we have a similarity with Maraviroc. But there's a lot of differences also between us and Maraviroc. Maraviroc first approval for HIV has a black box warning and toxicity, liver toxicity, which is very dangerous for the cancer patients who have their immune system is compromised. So our advantages is always a great thing to have. And as soon as we have that, Dr. Regner obviously will submit a synopsis of a protocol for stroke or maybe a full protocol, and he will use those data that Maraviroc has generated.
Yeah, and I mean, Chris will tell you there's a lot of different things that you can look at within stroke, whether it's fatigue or depression and, motor function return, sensory return. There's so many different things that we can look at to see the effects of leronlimab. Would you agree, Chris?
Absolutely, Scott.
Yeah.
It's focusing in on those patients and doing very accurate assessments of each of the endpoints that you would then go for at a later trial.
Yeah, and there's not much treatment for these patients, whether it's stroke or traumatic brain injury. So it's just a tremendous opportunity that Chris and I have been talking a lot about lately.
Right. When can investors start expecting dividends?
Once we have sales, and we talked about that a long time ago about giving dividends at the time that we have sales. Once we have sales and our cash flow is great and our stock is at the dream level, then definitely that's the time we will be thinking about that.
A viewer says, thank you for what you do. Do you plan on allowing compassionate use in the USA?
Well, for COVID-19, we're planning to submit a protocol for COVID-19 critical ill population, and then another protocol for compassionate for those who cannot enroll perhaps in that trial. Perhaps there is some inclusion criteria.
And the TNBC trial is studying leronlimab in combination with chemotherapy. How do we know the relative contribution of each? And couldn't it just be the chemotherapy providing the clinical benefit?
So I answered that already. I think that was the question that I had read. But go ahead to the next question, please.
Well, yeah, sometimes people pop on different times, but we can move on. What advancements do you see with the addition of the new board members?
Well, I'll tell you a few things, and Dr. Kelly can tell you more. Leash can bring us more visibility for HIV. And the data we have in monotherapy, nobody in the world ever seen anything like this. 7 years without any of the HIV drugs. It's just unheard of. And in regards to Dr. Haris Sitam-Raju, he believes this product has much more expanded application, and there are tests that he wants us to get involved in doing that could prove that to the scientific world. Obviously, we're making some room in the scientific world, but we need to make a lot more progress in that area. But Dr. Kelly, you want to add to that?
No, I mean, I think like we talked about before, we were very careful in who we chose, obviously, and we think we brought on probably the top immunologist in the world, as well as one of the top lung transplant physicians and somebody that deals with advanced lung failure, as well as Tanya's experience. So we think we have no doubt that we have chosen the right people moving forward.
Great. Does LL require special handling, and how long is the product's expiration date?
So as Dr. Ray indicated, there are up to 4 years of stability of the vials. And correct me, Dr. Ray, if I'm wrong. And in regards to the bulk product, it's in frozen negative 80 degrees Celsius. That has a stability of God knows how many years. So handling it has to be refrigerated. It can be at room temperature. I don't think more than 24 hours. Dr. Ray, am I right?
I think, Dr. Ray, you're on mute.
You're mute.
Okay. Yes, we have a long history of stability of this drug. Since we've been working since early 2000. With the current formulation, that is a commercial formulation, we have data that will support the final drug product in the vials up to 4 years, and that much data we have. And when it is frozen, it is stable for years. We have used frozen drug substance after 8 years. And at room temperature, we have stability data at room temperature. That is, we do every lot we put outside room temperature, and we have data up to 6 months. But after that, we see that that drug quality will start to deteriorate after 6 months. But we don't suggest that the drug is stored at room temperature. So always stored at 2 to 8 degrees before use.
Perfect. Thank you.
Thank you for that. Are there any plans to raise funds by having a warrant conversion incentive program?
We're looking at all those avenues and definitely we'll let the shareholders know soon if we do go in that path.
Are you now, and will you continue to use a leading public relations firm?
We are using a public relations firm right now, and we will always evaluate other firms that could help us.
And when are proxies for the annual meeting going to be mailed to shareholders?
So I think we just received SEC clearance to file the definitive proxy, and hopefully we will be doing that today.
Good. So on the last Emerging Growth Conference, Dr. Pourhassan mentioned the possibility of adding U.S. hospitals to the critical trial. Will this impact the start date of first critical patient being dosed for critical trial in Brazil?
I'm sorry, you talk about Brazil and then U.S. I got confused. Can you repeat that?
Yeah, sorry, I'm just reading the question. So on the last conference, you mentioned the possibility of adding U.S. hospitals to the critical trial.
Yes.
Excuse me. And will this impact? Okay. Will this impact the start date of the first critical patient being dosed for the critical trial in Brazil?
No, it will not impact that. If we do add that, that's just something that will happen in parallel, obviously.
Okay.
I think
and
we
on this
should
call,
should we go to the last question? Because I think we got to get Dr. Ray and Dr. Reckner and Kelly to get back to work. So let's take the last question, please.
Last question here. Okay.
Please.
Okay, from someone saying, I love your compassion and your integrity. I pray for you and your team to continue to move this drug to the finish line. So outside of the business aspect of CYDY, what's your biggest concern?
So first of all, thank you for your prayer. I always need it. My team needs it. And outside of CytoDyn, the question is outside of CytoDyn, what is my biggest concern?
Yes.
My biggest concern is always to keep myself aligned with Lord God. So when you ask me personal question, that's what my biggest concern is. So every night before I go to bed, I ask my wife, have I been good, my beautiful wife? And she says, yes, that's my biggest concern. Dr. Kelly knows about that because very often ask him, have I been good today, Dr. Kelly? And he always says yes, but sometimes he does say no.
I'm pretty tough on him, I can assure you.
Well, there's another question that might gear your answer. There's a lot of people saying that the FDA is controlled by big pharma. Do you fear this?
I never feared that, and I said that before. We are blessed to be I'm blessed to be in this country. I thank America for giving me the opportunity that I have. This country is an unbelievable country, the best superpower that has ever been around for the last 1,000, 2,000 years. You could never find a superpower like this. Their system is absolutely fantastic. There are flaws always in every system, but to say that the FDA, what they have done in the past 100 years, and keeping everybody safe in the United States as much as possible, and then compare it to the other countries, and you will see that you guys have unbelievable regulatory agency. And if there are corruptions here and there in certain big pharmas or even in some aspects and other government agency. Those are the things that happens with humans. So I don't mark FDA as a corrupt agency in any way, shape, or form. The best agency I ever seen or worked with, and I've been blessed to work with them. I'm not saying that because we're dealing with FDA, because I'm saying it because I do believe in that.
All right. Well, if you want to have any closing remarks, that was the last question for us, even though there's a lot more questions that we will be sending to you directly, so you can get back to work.
No, I appreciate it. Thank you for great moderating that you did. And the emerging growth have done a fantastic job giving us this opportunity. This company, I would ask shareholders to look at what we have done in 7 years and project with what we accomplished so far, what we're going to be doing in the next 6 months. By the end of this year, we hope to have answers for 2 breakthrough designations that we want to file for cancer. And we have documents and results and testimonies. Wherever that takes us, we will be continuing to getting it to the finish line. In regards to our COVID-19, we started this in the beginning of 2020, and now we are at the end of 2021, 2 years. But we are very close to some very important milestones: approval, emergency use authorization, Philippine sales. All of those are the things that were accomplished, and we are hoping to accomplish a lot more by the next 6 months and getting this product to the finish line. In regards to NASH long haulers, 2 new clinical trials that Dr. Reckner single-handedly brought it and did what he has done. I can't wait for him to do the same thing with Alzheimer and stroke and perhaps MS, as we talk about. But then the HIV, this is dragging for a while, but it opened so many doors for us because we are telling the public exactly what we get from the agency or from whatever we have. So the public can be 100%, the shareholders can be 100% aware of what we have. Because we're doing that, you might get disappointed when sometimes we believe that we're going to have something, but we don't get it. But we're still going to be honest and tell you what we have. Next 6 months, I believe it's very, very strong activities and events going to happen for us. So let's see what happens. Thank you for joining us in this call.
Oh, wonderful. Thank you, gentlemen, so much. Such an important technology that you all are developing over there. So thank you for joining us again on the Emerging Growth Conference, and we hope to see you again soon.
Same here. Thank you.
Thank you.
All right, everyone, stay with us. We have a few minutes of a break. We'll meet back here at the top of the hour for our last presentation. We'll see you soon.